Good morning, everyone. I'm Dr. Abhishek Pathre. I'm Assistant Professor in Pathology, Santosh Medical College and Hospital. Today, the topic which I'm going to take is Etiopathogenesis of a disease, cell injury, and degeneration. This is one of the important topics which we cover in pathology, and it is very interesting topic also. So the normal cell is confined to a fairly narrow range of function and structure by its state of metabolism, differentiation and specialization, and by constraints of neighboring cells and the availability of the metabolic substrates. Nevertheless, it is able to handle physiological demands, maintaining a steady state, and this steady state is called as homeostasis, and this is the basis of all the living organisms. Adaptations are reversible functional and structural responses to more severe physiological stresses and some pathological stimuli, during which new but altered states are achieved, allowing the cell to survive and continue to the function. When the stresses is eliminated, the cell can recover its original state without having suffered any harmful consequences. If the limits of adaptive responses are exceeded, a sequence of events follows that is called as cell injury. The cell injury can be reversible up to a certain point, but if the stimulus persists or is severe enough from the beginning, the cell suffers irreversible injury and it is called as cell death, which is the ultimate end point. The cell death, that is the end result of a progressive cell injury, a normal and essential process in embryogenesis and the development of organs and the maintenance of homeostasis. There are two principal pathways of the cell death. One is necrosis and one is apoptosis. These two topics are very important for the whole medical life, as the necrosis and apoptosis are the basic things which are going on in our body. The stresses of different types may include that induces the changes in the cells and tissues other than typical adaptations, cell injury and death in the form of intracellular accumulations and pathologic calcifications. Finally, the normal process of aging itself is accompanied by characteristic morphologic and functional changes in the cells. The types of stresses and stimuli which includes these are physical, which includes mechanical trauma, thermal, pressure, radiation, waveforms and electricity. The chemical, which includes organic as well as inorganic. The microbial, that can be bacterial, viral, fungal, parasitic and very so on. Immunological can be innate or adaptive. Nutritional can be undernutrition as well as overnutrition, and genetic can be inherited and sporadic. These are the types of stimuli or stresses that can happen to our body. So cell adaptations, which include hypertrophy, adaptation which I have already discussed, that is the changes which are happening in our body at a cellular level to compensate the changes from an external stimuli or internal stimuli. The cell adaptations include hypertrophy, hyperatrophy, hyperplasia as well as metaplasia. So hypertrophy refers to an increase in the size of the cells or cell organelles resulting in an increase in the size of organ. The hypertrophied organ has no new cells, occurs in organs and tissues where the predominant cell population is in G0 phase of the cell cycle. Very important. The increased size of the cells is due to the synthesis of more components of the cells. Hyperplasia, if we talk about, is an increase in the number of cells. Please focus. This is the increase in the number of cells, whereas in case of hypertrophy, that is increase in the size of the cells. So hyperplasia is an increase in the number of cells in an organ or tissues, usually resulting in increased mass of the organ or tissues. Atrophy refers to a decrease in the size of the cells. So when we say the organ has atrophied, it means the size of the cells or the organ has reduced. So atrophy refers to a decrease in the size of the cells or cell organelles resulting in a decrease in the size of the organ. Metaplasia is a reversible change in which one differential cell type, that is epithelial or mesenchymal, is replaced by another cell type. Mechanism of cell injury, that can be due to depletion of the ATP, mitochondrial damage, loss of calcium homeostasis, oxidative stresses, altered membrane permeability and endoplasmic reticulum stresses. These are the mechanisms of cell injury. So depletion of ATP causes reduced supply of impaired utilization of oxygen and the nutrients, and mitochondrial damage. Mitochondrial damage causes hypoxia, release of the toxins, and mutation in the mitochondrial genes that are the causes of some inherited disorders. So mitochondrial membrane transition or changes causes the changes in the influx and the outflux of the ions that hampers the formation of ATP. These are the mitochondrial matrix which showing when there is difficulty in formation of the ATP, which is the energy source of human body. And these changes in the mitochondria can ultimately, if not corrected, can lead to even necrosis when there is loss of membrane potential, inability to generate ATP, and when there is cytochrome C and other apoptotic proteins of production, that is called as apoptosis. This is again the mitochondrial damage causing the release of various enzymes, can be phospholipases, proteases, endonucleases and ATPases, causing the nuclear damage and the membrane damage. If we talk about the classification of the free radicals which are also involved in the damage to the cells at an organelle level, that can cause the electron transport chain, that hampering this electron transport chain, the lipid peroxidation, the phagocytosis and the nitric oxide. These are the free radical formation, and that is H2SO peroxide ion, and this causes the membrane damage of the organelles, most importantly the mitochondria. Again, this is a free radical formation. This is a graph showing free radical formation. Okay, membrane damage causing the phospholipid loss, lipid breakdown products, cytoskeletal damage. All this is caused by the free radicals which is generated in our body, and that can be reactive oxygen species causing the lipid peroxidation, phospholipid synthetase difficulty, and ultimately causing the membrane damage. This is again when there is ATP loss, multiple downstream effects happen. Mitochondrial damage causes leakage of the apoptotic proteins. Entry of the calcium increases the mitochondrial permeability and activation of the various enzymes. Reactive oxygen species activation causes damage to the lipids, proteins as well as DNA damage. Plasma membrane damage can be at cellular level and the enzymatic level, and ultimately the protein and misfolding and DNA damage can also happen. So the injuries can be reversible injuries and the irreversible injury. If the injury is reversible, that is the stimulus is gone, the cell function is retained at a normal level. However, if the injury is irreversible, that is the cell is not able to compensate with the injury, then biochemical alterations and the cell death can happen in the cell. Ultracellular or ultrastructural changes also happen, and these can be seen under the light microscope, called as light microscopic changes. And if it continues, finally the gross morphological changes can be seen in an organ with the naked eye. Reversible cell injury can be recognized under the light microscope as cellular swelling, hydropic changes or vacuolar degeneration, increased eosinophilia of cytoplasm and fatty changes. These are the reversible injuries, as you can see when the A is normal and the B is abnormal, showing the ballooning or the hydropic degeneration of the cells. This is electron microscopic view of the same. This is again the high power view under the electron microscope. Again the tubules of the hydropic degeneration under the electron microscope. So when there is damage occurs to the cell, there is bleb formation, intracellular volume of the fluid increases, mitochondrial swelling and calcification happens, disintegrated ribosomes and aggregated cytoskeletal elements can also occur. Dilated and vesicular endoplasmic reticulum can also be seen. Thank you.